Great, thanks very much, Nora. And thank you, Jeff and Carlos and the organizers of ILSI for inviting me here today. It's a real pleasure. So I'm going to be talking about using metabolomics to analyze food components, and I'll also be talking about some of the work that we're doing and others have been working on in terms of database development and making that information accessible to all. So traditional food component analysis is sort of highlighted through here, where there's often a process of collecting different foods, pure foods, fundamental foods, or foundation foods, or prepared foods, freeze drying, grinding, separating components through often liquid-liquid extraction. Sometimes there's further boiling to do further extraction, filtering, digesting, and then you put it through a variety of instruments to measure both the macronutrients and some of the micronutrients. It's been around for more than 100 years, processes, and this is how most food component databases have built. So there's one approach, which is the chemical analysis. And then, of course, people have published this information in various journals and books over the years. And so I'll be talking about both aspects today in terms of how we bring these two pieces of information into what we call electronic databases or open access web-based resources. And in some respects, they're both equally difficult. The chemical analysis obviously requires a lot of instrumental knowledge. Collecting data from books and journals is also tedious, but requires a fair bit of expertise. And it's almost essentially mining data to make it in essentially electronically accessible in data resources. Now, over the last century, a little more than a century now, more than 150 different food composition or food component databases have been developed. These are online. Many of them are based in specific countries, so they have regional focus, a regional flavor, analyzing the composition of the foods characteristic to those regions, cultivars or species, or foods that are commonly prepared in those regions. So there's a lot of data out there, but again, it's also highly scattered. Probably the best known, and in fact, the original food database, which I think appeared in the late 1890s, came from the USDA. So it's been rebranded as Food Data Central, and it now has a little over 400,000 foods cataloged in it. Vast majority of those foods are prepared or branded foods, things like chocolate bars or cereal, and then a much smaller set, what you call foundational foods, things like tomatoes and bananas and beef. The analysis is done through many of the methods that I just highlighted and continue to be refined, but depending on the food, you can get as little as 7 components to up to 90 different nutrients. Identified and quantified. And so that will cover things like carbohydrates, lipids, proteins, fiber, break it down into the specific fatty acids, minerals or metals, amino acids, and vitamins. Those are usually what we regard as the essential nutrients, and this is what we think of when we talk about nutrition, and to sort of view food as sort of a collection of between 50 to 90 specific compounds. Now, there are a few problems with today's food composition databases. In fact, there are 150 of them, many specific to different regions and countries, makes that distinct in different languages, different content, very different web or search capabilities, very different layouts. The tendency is to push for a wide variety of foods, tens to hundreds of thousands, but in terms of the number of compounds measured or quantified, it's typically in the dozens per food. Of course, there's the prepared foods, and I think people have found that this is one of the central weaknesses because everything has a different recipe, whether it's old Coke and new Coke and Diet Coke, or any variations on cereals or pizzas. And then the foundation foods, which are the components that make up all of those prepared foods. And again, that's a bit of a mix. The focus is on the macronutrients, a few of the micronutrients, but very little in most cases on sort of non-nutrients, some of the other newly discovered micronutrients. The components that contribute to aroma or flavor are often not measured or described. Contaminants, food additives are also generally absent from these things. Most of them use technology and techniques that were developed in the 1800s and early 1900s. And they don't really acknowledge the fact that the average food contains more than 10,000 detectable compounds. So typically, food composition database only provides 1% of the known or expected information about foods. Now, people have been trying to correct that. There have been efforts, the Dictionary of Food Compounds, which has been around for a while. Traditionally, it was a book. Now it's become a little more electronic. There's EFIS, which is the food additive database that's maintained by the USDA or FDA. There's Dr. Duke's phytochemical database, which originally was just a long list and has gradually over the years become a little more electronically friendly. And we reviewed a lot of the existing food constituent and food metabolite databases in an article from last year, just sort of talking about the strengths and the weaknesses and where things could be improved. And to some point, I think in that article and where a lot of people are moving towards is there is a need to have a bit of a rethink about how we measure foods and how we report that. We should be trying to exploit the advances in liquid chromatography, mass spec, GCMS, NMR, that's metabolomics, which we talked about 2 days ago, to identify and quantify many more components than the just 50 or so that are standardly measured. I think we also need to focus much more on the micronutrients, the non-nutrients, the contaminants, some of which can be used as food biomarkers, but also which seem to contribute a fair bit to our response to food, whether it's to flavor and taste, but also some of the more subtle effects relating to mood, or anxiety, or alterations in their gut microflora due to food. Additionally, I think we also need to look at how we design and present the data. A lot of the databases in food composition are not very modern. They don't have the sort of web-friendly or searchable capabilities that are typical of modern databases. We need to use more data mining, machine learning to help pull and extract data from both electronic and literature sources. We also need data to help with the analytical process. The food databases will have names, but they don't have structures. They don't have descriptions of the compounds. Some don't have very good information on the concentrations. If we want to identify these things, we need reference spectral data, NMR and mass spec. Very few of the databases, in fact almost none, have anything on the health effects or the flavor or aroma effects of these compounds. And most don't really have a lot of information on cultivar variants or strains. And so I would argue that we need to really focus on depth as opposed to breadth, and that we need to focus on the so-called foundational foods. So in terms of metabolomics for food analysis is sort of a simple illustration where we typically would go through an analysis where we would gather certain food samples, usually single foods, not the mixtures here. We go through extraction, and generally we try and convert things into some kind of liquid extract, not unlike what I showed in the first slide. The analytical methods used in metabolomics are NMR, GCMS, LCMS. Those have been around for a long time. Where the real developments over the last decade have been in the data analysis, being able to analyze complex mixtures, to be able to identify and quantify from thousands of peaks, individual components, and know exactly what they are, and then to interpret them in some biologically useful way. So in metabolomics, there are two routes. There's a targeted or quantitative approach, which uses the generally triple quadruple mass specs or NMR. You know what you're measuring. You have isotopic standards. You have accuracies or variations often less than 10%, are highly consistent, rigorously controlled. The other approach is untargeted, which is the more popular approach to metabolomics, and that's kind of the Wild West. We heard earlier about reproducibility. Untargeted metabolomics is not reproducible. It's highly qualitative. And it's one of the challenges I think that we face in the field of omics science, where when we're given two choices, we tend to choose the untargeted, qualitative, non-rigorous approach as opposed to the quantitative, rigorous approaches. And that's because the untargeted ones are great for discovery. They're great for getting a publication in Science or Nature, but as we are learning, a lot of them are irreproducible and wrong. So I'm going to talk a little bit about our efforts in terms of doing both food metabolomics on the experimental chemical side, and then our efforts as well to bring in information from what's known, what's in journals, books, and other electronic databases, and to consolidate them into open access resources that we maintain. So I'll be first focusing on the experimental parts that we do. So in my lab, we do targeted metabolomics. We've made a very deliberate decision, and because, especially if you're talking about food analysis, you need quantitative exact data of the components. This is an example of one of the kits that we do or run on food examples, food samples. We'll measure a whole bunch of fats. We'll measure biogenic amines, amino acids, and amino acid derivatives, fatty acids, organic acids. The list goes on. We've developed a variety of kits for food analysis. The kits, as I say, are more for our own use, but we're starting to distribute them to other labs. And some are for LC-MS methods, some use GC-MS, some are for NMR, some use inductively coupled plasma mass spec for metal analysis. They're all quantitative. Depending on the type of assay and the type of kit, you can measure between 20 to 700 analytes. We worked very hard over the last couple of years to become ISO compliant. Again, this is this issue of rigorous reproducibility, making sure that the numbers match time in and time out, both for the assays we run in our lab and then for the kits that we distribute to other people. This is an example of one where we're looking at animal foods. You can see lists of the amino acids and some of the organic acids, the fatty acids, the vitamins. But then there's also a variety of acyl carnitines that we'll measure, a variety of lipids and lysophosphatidylcholines, more sphingolipids, ceramides, cholesterol esters, diacylglycerols, triacylglycerols, more and more, and then the metals. So these are the things that can be measured in a single kit. So this represents hundreds of compounds that can be quantitatively measured to an accuracy of about 10% from sample to sample and down to nanomolar or sub-nanomolar levels. Plant foods are a little different. Again, you'll often find the same set of amino acids, but there are different organic acids, still some fatty acids. Vitamins are also tend to be more abundant. But because plants are different, there's quite a few more organic acids that we've added to the kits, other types of polyphenols and biogenic amines, a variety of terpenoids and sugar alcohols, and then contaminants, pesticides and herbicides that can be picked up in plant samples. And again, the metals, because plants tend to concentrate metals from the environment. So depending on the food type, depending on the situation, we'll have different kits for different samples. These are primarily GC, LC, MS, and ICP-MS measurements. So we've applied these techniques to a number of fruits and vegetables that are grown in our province in Alberta. Beetroots as an example. Described the number of compounds detected by NMR, 37, 26, well, I guess around 70 in terms of the GCMS, 38 metals, and then the mass spectrometry over 500. And then we've gone through a variety of other fruits and vegetables using these same multi-platform approaches to identify and quantify individual compounds in all of these locally grown produce. We've also moved into the analysis of other foods, particularly beverages, both milk, wine, and beer. In this case, we're using NMR. We're also using GC and LC-MS, but we've tried to automate the NMR approach. And many of the compounds that are of interest in wine and beer aren't so easily detected by mass spec, so NMR has been very useful. We've also used artificial intelligence, machine learning methods to help automatically deconvolute the spectra. And so something that would often take an hour can now take, in some cases, as little as 2 minutes to analyze. And this can be run over and over and over again. So hundreds of samples can be processed in a single day. These are some of the compounds that we detect in wine and beer, more compounds. So typically it's on the order of 100, and as I said, many of these things are not easily detected by other techniques. So a lot of our efforts have gone into at least experimental analysis of both expanding coverage, making things more quantitative, making more consistent, making things as automatic and as rapid as possible. So those are some of the analytical methods that we've been using to contribute to a variety of databases. And then we've also been looking at mining the literature. So I have a group sort of a little schizophrenic. We've got people on one side of the lab that do chemistry and another side of the lab that do computing. And some of the first databases we worked on were resources like Phenol Explorer. This is a collaboration with Augustin Scalbert, who used to be at INRA and is now at IARC. And this involved looking at thousands of reviewed papers, collecting the data by hand, getting information on about 500 polyphenols and covering 460 different plants. We've also looked at the polyphenol metabolites, gathered information on the pharmacokinetic parameters, and then also the effects of food processing on polyphenols. It's been a while since we last updated it, but this is one of our earlier attempts in a sense, food database. We've also worked with Augustin's collaborator, Claudine Menac, at INRA Clermont-Ferrand. This is PhytoHub with a focus particularly on phytochemicals as well as their metabolites, and covers about 350 different edible plants, fruits, and vegetables. We're helping them add a lot of physical chemical data, mass spec data, NMR data, more metabolites, and linking it to other databases to make this more extensive and more useful for the community, especially for people who are trying to analyze food, where reference spectra are really critical. Now, I think it's important to remember that microbes play a pretty important role in food. Fermented food is something that we've highlighted, talked about before, and its role for good health. Yeast is an essential fermenter. And we've been working on another database called the Yeast Metabolome database. And we were surprised to find that there's at least 16,000 different yeast metabolites. And given that yeast grows on many different things, produces a whole variety of transformation products. We've tried to link that to the proteins and enzymes that are responsible for these transformations. We've collected reference spectra, NMR, GCMS, and we've also used machine learning to predict many of these spectra. We've built out pathways, we've built out reactions, and we've built in extensive search similarity tools to help this, and for different needs and different people, and trying to make it useful to a broad community of users. I'm from Canada. Canada has legalized cannabis. I guess a few other countries are starting to do that. And we were asked to sort of build a resource to cover what is found in cannabis. And again, this has involved both literature mining and in-house analysis with the techniques and kits that I highlighted. So cannabis is complex. There's more than 6,200 compounds that we've been able to annotate. The general references usually say there's 500. That's wrong. We've also been able to get referential spectra, but we've also highlighted all of the targets that many cannabis compounds act on in the human body, because it's also a drug. We're hoping to wrap up the manuscript. The database itself is online and has been for a while. We are also working on natural products. NIH has asked us to build out a natural products database. This one focuses on collecting the NMR spectra. It's called the NPMRD. It was released earlier this year. It's a little different than some of our other databases because it supports archival deposition, and this is part of the effort that NIH and others are having for people to deposit their data when they publish. And so we're hoping to capture both NMR data and eventually mass spec data. Right now, the database has 1.2 million NMR spectra, but only a small number of which were actually deposited as FIDs, about 400,000 that were simulated from the known or assigned chemical shift, and about 800,000 using machine learned methods for calculating NMR spectra. We use a lot of AI and ML methods, that's artificial intelligence and machine learning, to help maintain and curate the database and check the quality of the data that's being deposited. We've not only analyzed wine and beer, we've been looking at milk. Characterizes a variety of different compositions of milk. This is commercial cow's milk, not breast milk or goat's milk, but specifically cow's milk. Interestingly, 2,300 compounds have been identified. About 70% of them have been quantified. We have a bunch of different concentration values for different forms of milk. And then we've also got MS and NMR spectra. Again, the tool is online. It allows you to support structural searches, spectral searches, text searches. All the compounds have detailed descriptions about what they do. The images, mole file, SDF files can all be downloaded. We've extended the work in milk to other livestock. And this is more focusing on meat and looking at their tissues. We're looking at both cattle, pigs, horses, sheep, and goats. And this represents stuff, again, that we've measured in our lab, as well as what's in the literature, using many different platforms, looking at different tissues and biofluids. The collection is smaller, it's about 1,200 metabolites if we exclude milk, but there's lots of data on concentrations. And the utility for this one has been less in food, but more for veterinary reference values, which is, again, interesting because the intent wasn't for that, but it's always a surprise to see where people end up using the data. Our major effort is the FooDB. This is something that we started more than 10 years ago. Currently, it's a database of about 71,000 compounds from 720 foundation foods. So these are largely the raw foods, the fruits and vegetables, meats, fish, livestock meat. And what we decided to do is sort of put our money where our mouth was, and to try and collect information on flavor, aroma, color, and human health effects for all these compounds. So there's about 5 million concentration values, covers 31,000 different text references. Only 1,400 compounds have really useful health effect information. About 2,700 have information on flavor and aroma. The others, we're still not sure what they do in our food. We've linked everything from the compound descriptions to the food types and images of the food. We've made things as searchable as possible from text, from food and food type, and species, by chemical name, chemical structure. But you can also search by spectra as well. So you can browse the database as well as through search, and people can browse at the food level. We have what's called a food card that provides detailed descriptions of all the food items, images, the taxonomic classification. And then within each food, the food-specific chemical composition and concentration data. Although not every compound has concentration data, but at least it tells you what's known or has been detected through things like untargeted methods. So in the food view is one approach, the chemical view is the other one. So these are compound cards as opposed to food cards. And again, people can browse through the chemical version of it. And this way you get the pictures of the structures, you get the physical chemistry data, the mass spec, NMR spectra. But this also captures information about the health effects, the flavor, color, and aroma data that's been either collected or published. A lot of that data has to be curated, pulled in through a variety of techniques. And again, we have to use text mining, text recognition, or machine learning techniques to help build that out. It's a variety of search options. People can draw structures or partial structures and look for similar molecules. They can search by text. They can search by spectra. A whole variety of other ways of accessing and viewing the data. But there's still a lot to do. It's been around. It's been used a lot. We're obviously working on improving the interface, speed, usability. We'd like to add more filters to allow more compatible queries for people that are in the food and nutrition science, so that you could actually look for foods with the highest content of, say, oxalic acid or the lowest content of phytic acid. We're also trying to standardize some of the structure rendering. It's a bit of a wild west in terms of how people both draw their structures and how they name their structures. We're improving the NMR and MS spectral quality. We're introducing a new ontology for chemistry called ChemFont. It's called the Chemical Functional Ontology. similar to the gene ontology, which some of you may be aware of. This will make food data and chemical data much more machine readable, much more mineable. And so that's allowing us to add a lot more data to the database. We hope to wrap it up in October. So that's about 10 years after we first started it. And we've written up a good portion of the manuscript and hope to submit it in October. So the question is, and you know, where to go next? I think it's important that when we talk about food analysis, it's not just about macronutrients, it should be about micronutrients. And then that means embracing some of the technologies that are in targeted quantitative metabolomics. I notice that I'm excluding untargeted because I think we still need that information about concentrations because that's very important. Dosing, if we think of food as drugs or as nutrients as drugs, we need to know how much is being added. I think we need to start incorporating metabolomics data into these food component databases. We need to make them more modern, more interactive, use tools that are now available in many web systems, from JavaScript to cascading web design techniques. We need to appeal to a broader community. The information about food constituents shouldn't be just for nutritionists. It should be for consumers. It should be for physicians. It should be for chemists. It should be for people looking for biomarkers. So capturing information about flavor, aroma, health effects, and making it accessible might help address some of these issues that we have with communicating science to the public. I think we need to focus much more on the foundation foods. This is where the periodic table of food initiatives is, I think, a paramount importance, because they're trying to look at the foods from around the world, trying to capture strain and cultivar differences. And by understanding what's in our foundation foods, then we can better understand what's in the prepared foods. We also need to think about measuring chemicals from food processing, cooking, and frying, preparation, along with the food contaminants that often arise from preparation or what are added or inadvertently added to our food, whether it's in the field or in the preparation facilities. I think we also have to embrace developments in machine learning and artificial intelligence to continue to update and annotate and expand what's in these composition databases. So I think there's a lot of modernization that needs to be embraced, and hopefully some of the things I've described are our first steps to doing that. So with that, I want to thank some of our funders and our collaborators, including Lorraine Brennan, who will be speaking after me. Thanks very much.
Thanks, David. Great talk. Any questions? Please, just there. Yeah.
Wow, it's kind of amazing one lab could accomplish so much. So thank you for this contribution. Yesterday, you were saying that there's the possibility of some more persistent biomarkers to reflect what people have eaten than some of the bioactives with fairly short half-lives. What are those? How are you going about finding them and excluding the ones that are homeostatically regulated to be constant in the blood, like minerals from bone, if you don't get it from the diet?
Yeah, I mean, part of the interest in looking at what's in the food gives us, I guess, a hint of what should be biomarkers of food intake, or BFIs. And
so more consistent food intake.
That's right.
Rather than a
But I think there's two types of things that we're looking for food biomarkers. So there's the transient ones that are in your bloodstream or urine for 12 to 24 hours. I think there are also ways of tracking biomarkers that are more essentially habitual food consumption. And that might be looking at what's in hair. And we've looked at that, and actually it forms a lot of information about nutrition, fingernails, bone and teeth samples. Adipose tissue is another way of getting much more information about habitual consumption or prior consumption of foods from further back. The best food biomarkers are ones that are essentially inert to transformation. Just like some drugs like metformin pass right through you with no transformation. Some of the best food biomarkers are things like proline betaine, which does not get really metabolized at all, and it's an excellent biomarker for citrus consumption. And 3-methylhistidine, 1-methylhistidine also are inert to metabolism. They're great biomarkers for certain types of meat. And as I think we've published through the FooDB Consortium, which is a European initiative led by Edith Veskins, about 20 different papers describing high quality food biomarkers for most of the major food groups and food types. So that's out in the literature, and that's in some of our databases as well. So one is called MarkerDB, which lists a lot of those food biomarkers. And then Exposome Explorer is another resource we've worked on that also captures food biomarkers of consumption.
Please.
Thank you. You touched on looking at different strains and cultivars for plants, and I just wondered, when the database is first published, is the user going to be able to get a feel for what is a sort of relatively consistent constituent of a food, and what is something that can vary very markedly depending on things like season or geographical location?
Yeah, and I think this is our effort with FooDB is basically to capture the generic apple, the generic peach, and the generic carrot. And the PTFI initiative, which you'll hear a bit more about, is to look at those seasonal variants and cultivar variants. That's going to require much more data collection. Our efforts, as I say, just from one lab, self-financed basically. But PTFI has much more in terms of resources to collect these different samples and to analyze them through different labs around the world. So that will hopefully show up in a few years in the PTFI databases.